on, listen please. The pollen grains structure, internal structure, general account already learned. Now it is something about pollen grains. They are pollen viability, pollen products, pollen allergy, pollen bank. So what do you mean by that? The period of pollen grains that remain functional, the period of pollen grains that remain functional is called pollen viability. The activity of pollen grains, the tendency of germination, which are all depending upon the nature of the pollen grain. And so the duration or the period or the span of pollen grains that remain functional is called a pollen viability. Pollen viability means, you know, in wheat, paddy, like grasses, the viability of pollen grains, the viability of pollen grains is only for 30 minutes. Only for 30 minutes they remain functional. After that, they will not germinate when the pollen, after the pollination, if takes place. Whereas the members of family like Rosaceae, Leguminosae, Solanaceae, etc. The viability of pollen grains will be for months. They remain functional for months, several months. Not several months, 1 or 2, maximum 2 months. 1 month, within 1 month, they remain functional, otherwise it will dry off. See, the period of pollen grains that remain functional is called a pollen viability. Example, in wheat, paddy like grasses, the viability of pollen grain is only for 30 minutes. And the second example, the members of families like Rosaceae, Leguminosae, Solanaceae, the viability of pollen grains will be 4 months. 4 months, maximum 1 month, 1 or 2 months. Okay. And that is about pollen viability. Then pollen products. pollen grains are nutritional. Not all the pollen grains. The pollen grains are nutritional. Hence, some of the pollen grains are edible, eatable. Some pollen grains are used as syrups, drugs, etc. Bee pollen, example. Some pollen grains are used to make syrups and drugs, hence they are medicinal. So, medicinal. Some pollen grains are used to make syrups, drugs, etc. Hence they are medicinal. Example, bee pollen. Then the pollen grains of some plants are used by athletes and race horses for better performance. See something about pollen grains. Generally, pollen grains are very small and they are nutritional also. Hence, some pollen grains are edible. They are taking as stimulant, we can say. Some pollen grains are used to make syrups, drugs, etc. Hence, they are medicinal. Example, bee pollen. Bee pollen tablets will be there. It is highly expensive and moreover, it is used as stimulant and for the body health. Okay. The pollen grains of some plants are used by athletes and race horses. So those horses participate in horse race. Their muscular activity will be stimulated by some pollen grains in western and eastern countries. See, the pollen grains of some plants are used by athletes and race horses for better performance. And so these are all something about the pollen products, drugs, syrups, bee pollen, etc. Okay. Then pollen allergy. So formerly I told you the pollen grains are very small. They have the minute outgrowths, and some are sticky. Anyway, these minute pollen grains will be passing into the lungs through the inspiratory gases or inspiration. As it is very small, it will make irritation to our respiratory tract, and that may lead to the coughing, snuffing, so and so, etc. And it will lead to the asthmatic irritations or bronchitic irritations, and that is why they are allergic. See, for example, the pollen grains are small and maybe sticky or spinous. Some pollen grains are allergic or asthmatic or bronchitic, etc. Example, the pollen grains of carrot grass or parthenium imported from America with wheat grains. The pollen grains of carrot grass or parthenium imported from America with wheat grains will be allergic to man. See, some pollen grains are allergic, asthmatic or bronchitic, etc. So you know, the pollen grains of carrot grass or parthenium, which will be imported from America with the wheat grains, knowingly or unknowingly, will be allergic to man. It will cause asthma or bronchitis, etc. And that is why the majority of pollen grains are not allergic, but these types of plants that producing small pollen grains which will irritating to the respiratory tract and leads to the bronchitic or asthmatic processes. See, that is about pollen allergy. Then pollen bank. So you know, pollen grains are actually used for cross-pollination, which is a part of crop improvement program. So qualitatively and quantitatively high yielding plants the pollen grains will be very important for doing cross pollination. It is a crop improvement program because new varieties of plants can be easily produced by cross hybridization. That you can see later, the plant breeding. The breeders breed new breeds which are qualitatively and quantitatively high yielding variety of plants. So such pollen grains are collected and stored for further use. So it will cryopreservate, just like the semen collected from animals, which is used for herd improvement program or artificial insemination. And like that, the pollen grains of qualitatively and quantitatively high yielding plants are collected and stored in minus 196 degrees centigrade at liquid nitrogen in a frozen state, and then it is transporting various parts of the world for crossbreeding if needs. And so such process is called cryopreservation. Just like a collection of sperms or semen which will be stored for further uses. So we can say such process is called a pollen bank. The storage of pollen grains is needed for breeding purposes. The breeders breed new breed plants or hybrid plants that are qualitatively and quantitatively high yielding varieties. So the storage of pollen grain is needed for breeding purposes. The breeders breed new breed plants or hybrid plants that are qualitatively and quantitatively high yielding varieties. The process of storage of pollen grains in minus 196 degrees centigrade in liquid nitrogen in a frozen state for further use is called cryopreservation. Name is not given in your textbook. Cryopreservation. It is just like semen preservation of animals for artificial insemination. See, so storage, blood bank, storage and exchanging the blood if deficiency or deterioration happens for the patients. Like that, crossbreeding nowadays a better program for the crop improvement of better yielding plants. See, the storage of pollen grain is needed for breeding purposes. Various plants nowadays, what is the era of cross variety plants production and animals for man's use. The breeders breed new breed plants or hybrid plants that are qualitatively and quantitatively high yielding varieties. The process of storage of pollen grains in minus 190 degrees centigrade in liquid nitrogen in frozen state for further use is called cryopreservation. It is just like the semen preservation of animals for artificial insemination. It is a part of crop improvement program. Crop improvement program. Okay. And so that is about pollen viability, pollen products, pollen allergy and pollen bank. Remember please, pollen viability, the period of pollen grains that remain functional for pollination and further development. Pollen products, as the pollen grains are of some plants are highly nutritive, they are edible most probably in western and eastern countries. In India there also. Example, you know the pollen grains of Hibiscus rosa sinensis or China rose is edible for those who are suffering the imbalancing of hormones. Nowadays it is used. Some tablets will be produced from the pollen grains of Hibiscus rosa sinensis belong to the Malvaceae. Some of the members of Malvaceae, the tablets are used, tablets are produced from the pollen grains, which will useful for imbalancing of, useful or remedial, useful for what? The imbalancing of hormones, especially the estrogen hormones. Then pollen allergy, parthenium. Remember please, parthenium, that is carrot grass, highly irritating those who are suffering from asthmatic or bronchitic allergy. You know, it causes allergy to man. So it was actually imported from America through when the wheat is imported. So the grains will be together with the wheat grains, these seeds of these plants or carrot grass was also there, and now it is omnipresent in India, and it is actually allergenic. Then pollen products, you know, syrup, drugs, bee pollen, which are the products obtained from some pollen grains of the plants. And moreover, it is a stimulant. Some pollen grains are stimulant to those performing different kinds of activities. Say, runners, race horse, they are all used the stimulant. It is actually medicinal cum stimulant for the better performance of the pollen bank. The storage of pollen grains is needed for breeding purposes. The breeders breed new plants or hybrid plants that are qualitatively and quantitatively high yielding varieties. The process of storage of pollen grains in -196 degrees centigrade in liquid nitrogen in frozen state for further use is called cryopreservation. It is just like the semen preservation of animals for artificial insemination. Then the pollen bank process is a part of crop improvement program. Better pollen grains will be stored for future purposes. Okay, and so that's about pollen viability, pollen products, pollen allergy, pollen bank. Carpel, ovule, embryo sac, or megasporophyll, megasporangium, female gametophyte. See, gynoecium is the female reproductive part of a flower. Gynoecium, which is formed of simple units called carpel. Each carpel contains a solid free end which is called stigma. This is stigma and a slender style and a swollen ovary. Ovary has an ovary wall enclosing numerous ovules. Ovary becomes fruit. Ovule becomes seeds after fertilization. This is ovary, this is ovule. This is thalamus. This is stalk. From here ovary, stigma style ovary. This is parts of carpel. The parts. Stigma is the free end of carpel. It is the landing platform of pollen grains. Usually sticky. See, it may be branched or unbranched. Most probably unbranched. Something about stigma. Stigma is the free end of carpel, maybe solid. It is the landing platform of pollen grains. Pollen grains are deposited, or the receptive end of pollen grains will be the stigma. Usually sticky in nature. It may be branched or unbranched. That is about the stigma region. Style is a slender part that connects the stigma and the ovary. Maybe tubular. See, this is style connecting stigma to ovary. Then ovary is the basal swollen part of carpel. Ovary has an ovary wall. Ovary wall, pericarp, fruit wall, pericarp. Ovary wall becomes fruit wall, the pericarp. Pericarp is the protective covering of fruit, a simple fruit. Fruit wall is called pericarp, having epicarp, mesocarp, endocarp. Ovary becomes fruit and ovary wall becomes the fruit wall. See, ovary encloses ovule. Ovary encloses ovule or ovules. After fertilization, the ovary becomes fruits. And ovule become seeds. Ovary is resting on the thalamus because thalamus is the stout tip part of pedicel, the flower stalk, on which the floral parts are arranged or all the floral parts are produced. See, these are all about the megasporophyll. Each megasporophyll, the carpel contains 3 distinct regions, namely stigma, style and ovary. You already learned in the morphology also. Stigma is the free end of carpel, which is usually solid. It is the receptive center or landing platform of pollen grains, usually sticky in nature. It may be branched or unbranched. Style is a slender part that connects the stigma and ovary. It may be tubular. The basal solen part is ovary, which has an ovary wall. Later becomes pericarp, and the ovary encloses ovules or ovule, and after fertilization, the ovary becomes fruit and ovule becomes seed. See, and so ovary, style and stigma constitute the parts of a carpel. So monocarpellary, bicarpellary, tricarpellary, pentacarpellary or multicarpellary, syncarpous or apocarpous conditions already learned in your morphology. If a single carpel is there, it is called monocarpellary. If 2, bicarpellary. If 3, tricarpellary. If 5, pentacarpellary. If more than 1, multicarpellary. If they are all united to form a single ovary, called syncarpous condition. And if in a multicarpellary condition, if the carpels are free from one another, it is called apocarpous condition. Example, rose. See, apocarpous condition already learned in our morphology of flowering flower. So that is about carpel, stigma, style, ovary. Next is ovule or megasporangia. Megasporangium or ovule. Ovule is an oval body. Developed from the placentum of ovary. Ovule is an oval body developed from the placentum of ovary. Stalk of ovule. Stalk of ovule is called a funicle. Stalk of ovule is called a funicle. The proximal end of funicle is connected to the placenta. Placentum is the soft tissue. Placentum is the soft tissue from which ovules are produced. Distal end of the funicle is connected to the body of ovule. Tissue or body of ovule. Tissue of ovule is called nucellus. Covered by 2 layers of membrane. 2 layers that you can see later, integuments. See, megasporangium or ovule that later becomes the seed after fertilization. So the ovule is an oval body. This is an oval body. Developed from placentum of the ovary. The stalk of ovule is called funicle, and this is stalk. This is body of ovule. This is stalk. Stalk or stamen is filament. That of ovule is funicle. This is the proximal end. Proximal end is connecting to the placentum. Placentum is the soft tissue seen in the ovary or ovary wall from which the ovules are produced. The distal end of the funicle is connected to the, this is distal end. Here you can see a connective, and this is connecting to the body of ovule, and this part is called hilum. See, the point of attachment of, see, the point of attachment of funicle and called hilum. It is just like a connecting part. See, the point of attachment of funicle and body of ovule. This is the body of ovule. This is funicle. Point of attachment of funicle and body of ovule is called hilum. This is general account. This tissue is called nucellus. Nucellus, protective covering, that is after the formation of seeds, it becomes testa and tegumen, outer integument and inner integument. 2 layers of covering is there to connect the mass of tissue called nucellus of the ovule. So the nucellus is the tissue of the ovule which is connected, covered and protected by two layered covering called outer and inner integuments, which later becomes the testategument part of seed coats. So ovule becomes seed, then covering becomes seed coat, and two layers of covering is there. And see, that is general account of megasporangium. Here also ovule, the most important part of the carpel, where inside the embryo sac is developed, which is developed within the ovule, there the nucellus, and so from the nucellus of the ovule the embryo sac is developed, which is the most important part of ovule, where inside the egg, the female gamete is produced, and that is why it is also known as female gametophytes. Female gamete producing tissue or structure, female gametophytes. Okay, then megasporangium produces megaspores. Megasporogenesis. Just like microsporogenesis in male parts. Megasporangium producing megaspore. Megasporangium producing megaspore. Ovule producing the embryo sac. How? And how the development happens in them. The formation and development of functional megaspore from diploid megaspore mother cell after is called see, the formation and development of functional megaspore from diploid megaspore mother cell or MMC after meiosis is called megasporangium. So we need functional megaspore. 4 megaspores are formed, among which what is functional? And that functional megaspore develops into female gametophyte, where inside the female gamete or egg is produced. And so we know the formation and development of functional megaspore from diploid megaspore mother cell after meiosis is called megasporogenesis. So formation and development of microspore from microspore mother or pollen mother cells after meiosis, that is called microsporogenesis. Here it is megasporogenesis. Resultant product of megasporogenesis is functional megaspore. Otherwise, we can say, or in Indian you can say, the microsporogenesis results the formation of microspore or pollen grains. So formation of pollen grains, microsporogenesis, formation of megasporogenesis. Then how and what development takes place from megaspore mother cell to the formation of megaspore, functional megaspore or embryo sac. Suppose ovule is an oval body. At the free end of this tissue, a cell become prominent. That means this is nucellus tissue. This is funicle. This is anovule, constitute anovule. This is hilum. This is, namely, in a true sense, it is called a archisporial cell, which is later modified as megaspore mother cell. This is see, this particular cell is called a archisporial cell and later modified as megaspore mother cell or MMC, which is diploid. So, taking this MMC, megaspore mother cell, it is diploid. It undergoes 3 successive division, resulting 8 nuclei within the, before that, it undergoes meiosis. Reduction division takes place. RD stands for reduction division, forming 1, 2, 3, 4 cells. So within the ovule, one, two, three, four. These are all called megaspores. They are haploid. So in some place, at the free end of the ovule, especially at the nucellus, a cell becomes prominent which is called archisporial cell, that later modified as megaspore mother cell. Modified as megaspore mother cell. And this megaspore mother cell undergoes meiosis or reduction division, resulting in the formation of 4 megaspores arranged in linear manner. That is called a linear tetrad. Linear manner, one by one. So, bilateral, decussate, T-shaped, linear tetrad, tetrahedral tetrad, the various types of spore tetrads. Means, points remember, bilateral, decussate, T-shaped, linear, then tetrahedral. See, once again please, the nucellus and ovule carrying nucellus, the tissue, at the free end an archisporial cell which is modified as megaspore mother cell or MMC. It is diploid, and this cell undergoes reduction division, forming 4 haploid cells, the megaspores arranged in linear manner, and that is why it is called linear tetrad. 3 of them disintegrates, and 1 remain functional and enlarge in size, which is haploid. It is called functional megaspore. Functional megaspore. Then megaspore is formed. And what about the formation of embryo sac? So listen. The functional megaspore undergoes 3 successive division. The nucleus divides into 2. First division. One, two, three, 4. Second division. And moves towards the periphery, opposite poles. Third division. So, the nucleus of the functional megaspore divides 3 successive divisions, resulting in the formation of 8 nucleated condition. 8 nucleated condition. So the functional megaspore enlarges in size and becomes female gametophyte. See, next line. 3 among them remain there at the opposite poles. One nucleus comes to the center from the poles. So 3 plus 3 plus 2, 8 nucleated condition. It is going to be the female gametophyte. And so here also the disintegration, disintegrated cells, and wall formation takes place. Three wall formation takes place. And now this is female gametophyte. Or embryo sac. Female gametophyte or embryo sac. And here you know and the disintegrated region gets ruptured. Final stage. See the functional megaspore, 3 successive division, 1, 2, 3, resulting 8 nucleated conditions, 4 each on the opposite poles. As per the production of number of nuclei, the megaspore becomes large in size and it becomes the female gametophyte. So 8 nucleated condition is there, among which 3 each remain on opposite poles of the embryo sac or female gametophyte, and wall formation takes place. 2 nuclei from opposite poles remain there in the center, that is the nuclei from poles, hence polar nuclei, having no wall formation around them, and 3 cells at the opposite poles and a large single cell in the middle carrying 2 cells, 2 nuclei. So this is ruptured region. Small opening. Outer integument, inner integument, eggapatus, nucellus, embryo sac, polar nuclei. Central cell, antipodals, chalasa, hilum, These are the parts of a typical, these are the parts of a typical These are the parts of actually a ovule. So we know this ovule becomes this states this ovule. This ovule becomes this is the opening is called micropyle. Protective covering is indicuments. Okay, listen please once again. So this was the ovule. Archisporial cell is developed. It is modified as megaspore mother cell or MMC. And the megaspore mother cell undergoes reduction division, resulting in the formation of linear tetrad, 4 megaspores, among which 3 disintegrated and 1 becomes enlarged. That is called a functional megaspore. And development of functional megaspore up to the formation of embryo sac. These are disintegrated cells, enlarged in size. That is functional megaspore. This one. Three successive division. 1, 2, 3 successive division resulting 8 nucleated. 4 each on opposite poles. 3 remain there at the poles. 1 each come to the center from the poles. Followed by wall formation takes place, engulfing a nucleus within the cell. Whereas centrally located 2 cells remain there, and which is kept inside the large cell, the central cell. And so this is the embryo sac, female gametophyte or embryo sac. This disintegrated region becomes cellulose, so we know this is the minute opening. This is minute opening by rupturing the indicuments, outer and inner indicuments. So this is micropyle. This is micropyle. The outer and 2 indicuments, outer and 2 indicuments, inner indicuments, thus becomes 2 indicuments. So this is the micropyle. This is the opposite region. The region of embryo sac at the micropyle end, within that there are 3 cells constitute egg apparatus. An egg plus 2 synergids. An egg plus 2 synergids giving egg apparatus. Nucellus, the tissue of the ovule, nucellus. This is embryo sac. Centrally you can see 2 polar nuclei, 2 in number. This is the central cell, keeping 2 nuclei, binucleated cell. Antipodals at the chalasa end. Chalasa is the place opposite to the micropylar end. That is the base of the ovule, chalasa. Chalasa end, you can see 3 cells which are called antipodals. Then hilum, point of attachment of ovule. Then funicle, the stalk of ovule. This is an orthotropous ovule. Here you know this is the funicle, micropyle, which are all on the same line, which is called orthotropous ovule. So this is the proper formation. And anatropous ovule, here you can see the funicle and micropyle are come near to one another, making the ovule anatropous. So listen please, an anatropous ovule. See here, it is the anatropous ovule. This is orthotropous ovule. In your, the development will be seen or expressed in your textbook as the orthotropous ovule, but the figure given, final figure is given the anatropous. What is the difference between anatropous and orthotropous and anatropous ovule? In orthotropous ovule, the funicle and the micropyle, which are all on the same line, whereas here in the anatropous ovule, the micropyle and funicle is coming very near to one another, very close to one another. This is ortho. This is orthotropous. This is hemianatropous. This is anatropous, ambulotropous, circinotropous. Different kinds of ovules will be there. To remember, points to remember. This is anatropous ovule. No, orthotropous ovule. This is hemianatropous. This is camphilotropous. This is different kinds of ovule. Points to remember: orthotropous, micropyle and funicle on the same plane. Anatropous, micropyle and funicle very near to one another. Hemianatropous, at right angle to funicle, the micropyle. Here, not at a right angle, but inner to the hemitrichous. This opening is coming very near to the, not so near, very near to mustard. Then circinotropous, ginger, etc. So remember, please, this is the funicle is opening is there, but 360 degree carving is there. So this is circinotropous. It is not needed for your examination, but to remember, orthotropous, anatropous. This is orthotropous, this is anatropous. Development is seen as orthotropous and the final figure is given as anatropous. This is anatropous, second one. And here direction is opposite, so that funicle and micropyle is very near to one another, hence it is anatropous ovule. And that is why we can say the formation and development of diploid Formation and development of functional megaspore from diploid megaspore mother cell after meiosis is called megasporogenesis. Megasporogenesis. And structure of ovule and some key points in connection with the ovule. Okay, see, micropyle, outer and inner integuments, then embryo sac, chalasa opposite to the region, chalasa, micropylar end. 3 cells, 2 synergids and an egg cell, which is collectively called egg apparatus. So central tissue, nucellus. Central 2 nuclei, polar nuclei coming from the opposite poles. Embryo sac is there. That is the female gametophyte. The central cell. So that is central cell carrying 2 nuclei. Then antipodals, 2 antipodals, the chalasal end. This is chalasal end. And so that is about micropyle, integuments, synergids, egg, nucellus, polar nuclei, embryo sac, central cell, antipodals, chalasa. So the embryo sac contains 3 cells each on opposite poles, 3 plus 3, that is 6. Then a large central cell carrying 2 nuclei, dikaryotic, that is a single cell. So 3 egg apparatus, 3 antipodals plus a central cell constitute 7 cells. So 7 celled 8 nucleated condition is there in a typical embryo sac of angiosperms. See, about mature ovule. Points to remember. Tissue of ovule. Nucellus. Protective covering of nucellus. Integuments. Stalk of ovule. Funicle. Point of attachment of funicle to ovule. Hilum. Base of the ovule. A minute opening at the integuments. Micropyle. region of ovule opposite to the micropyle. Chalasa. Embryo sac. Largest cell in the embryo sac, that is central cell. Binucleated cell in the embryo sac. Components of egg apparatus. Egg cell. Components of egg apparatus: 2 synergids and an egg cell. See, listen please. About mature ovule, once again please. These are all entrance questions. Moreover, in theory, it is for 1 mark questions. Tissue of the ovule, nucellus, protective covering of the nucellus, integuments, stalk of ovule, funicle, point of attachment of funicle to the ovule, hilum, base of the ovule, chalasa, the minute opening at the integuments, micropyle, region of ovule opposite to the micropyle, chalasa, largest cell in the nucellus, embryo sac, largest cell in the embryo sac, central cell, binucleated cell in the embryo sac, central cell, bikaryote, binucleated or dikaryote, dikaryotic cell in the embryo sac, central cell. Components of egg apparatus: 2 synergids and an egg cell. 3 cells in the embryo sac at its chalazal end, antipodals. 3 cells in the embryo sac at its micropylar end is egg apparatus. 3 cells in the embryo sac at the chalazal end, antipodals. That of micropylar end, egg apparatus. Mature embryo sac is 7-celled and 8 nucleated. 7-celled and 8 nucleated. Okay. Female parts, the male gametes will be produced after pollination during fertilization. Female gametes get ready for receiving the male gametes from the pollen grains. So pollination which is an intermediate process for fertilization in plants. So pollination. the transfer of pollen grains from anther to the stigma. Transfer of pollen grains from anther to the stigma is called pollination. So what is pollination? The transfer of pollen grains from anther to the stigma. So this is the stigma and this is the anther. Here pollen grains are produced. This pollen grain from the anther is deposited either directly or by the agents. Anyway, the transfer of pollen grains from anther to the stigma is called pollination. Pollination is of two types, namely pollination is of two types, namely self-pollination and cross-pollination. Self-pollination or cross-pollination. Self-pollination and cross-pollination. So cross-pollination is also known as allogamy or xenogamy. Allogamy or xenogamy. Self-pollination is of two types. Pollination is of two types. Self-pollination and cross-pollination. Cross-pollination is called allogamy or xenogamy. You can say it. First is self-pollination. Self-pollination. The transfer of pollen grains from anther to the stigma of the same flower or stigma of that of another flower of the same plant. The transfer of pollen grains from anther to the stigma of the same flower or that of another flower of the same plant is called self-pollination. See, what is self-pollination? The transfer of pollen grains from anther to the stigma of the same flower or that of another flower of the same plant. Suppose this is a plant. So it produces beautiful flowers. See, these are all flowers. Transfer of pollen grains from anther to the stigma of the same flower or that of another flower of the same plant. See, that of another flower of the same plant. So this is the pollination takes place in the same plant, that is self pollination. Self pollination is of two types. Self pollination, that is autogamy and geitonogamy. Autogamy and geitonogamy. See, transfer of pollen grains from anther to the stigma, that is pollination. Transfer of pollen grains from anther to the stigma is pollination. In the same flower or another flower of the same plant, these two constitute the self-pollination. So here, self-pollination is of two types, that is autogamy and geitonogamy. Autogamy, pollination in the same flower. Transfer of pollen grains from anther to the stigma of the same flower, that is autogamy. Or transfer of pollen grains from anther to the stigma of another flower of the same plant is called geitonogamy, that is pollination in different flowers of the same plant. Flowers of the same plant. Plant is same, flowers different. If it is in the same plant, it is autogamy. Or if in the pollination takes place in different flowers of the same plant, it is called geitonogamy. So geitonogamy, here an agent is needed. Thus, an agent-oriented self-pollination is geitonogamy. To another points. See, here we can say the transfer of pollen grains from anther to the stigma of the same plant, that is autogamy. Here, transfer of pollen grains from anther to the stigma of another flower of the same plant, that is called geitonogamy. Both autogamy and geitonogamy constitute the self-pollination. So listen, transfer of pollen grains from anther to the stigma is called pollination. Pollination is of two types. First is self-pollination. Second is cross-pollination or allogamy or xenogamy. The transfer of pollen grains from anther to the stigma of the same flower or that of stigma of another flower of the same plant is called self-pollination. Self-pollination is of two types, autogamy and geitonogamy. Transfer of pollen grains from anther to stigma of the same flower is autogamy. Transfer of pollen grains from anther to the stigma of another flower of the or different flowers of the same plant is called geitonogamy. And so, to say shortly, autogamy is pollination in the same flower. Geitonogamy is pollination in different flowers of the same plant. Here, an agent is needed. Thus, an agent-oriented self-pollination is geitonogamy. See here the figure, listen to the figure. A plant with numerous flowers. Pollination takes place here. Here the pollination takes place, autogamy. So from this flower to this heteronogamy, this to this heteronogamy, but the agent is needed. Otherwise no heterogamy takes place, though it is self-pollination. Then second one, this is first. Second is cross-pollination or xenogamy or cross-pollination, allogamy or xenogamy. +2 level. So you can go through cross-pollination, allogamy, xenogamy. So the transfer of pollen grains from anther to the stigma of the flower of another plant, flower of another plant of the same species. Plant may be genetically different. Genetic variations, genetically different plants, this process will be going on. So crossbreeding or allogamy or xenogamy, which are all the part of crop improvement program, or we can say a part of green revolution, in order to produce qualitatively and quantitatively high yielding varieties of plants can be produced through this method. So disease resistant capacity, prevent the insect pest or yielding capacity greater, early production of seeds, then flowers, etc., which all the benefits of new plant or hybrid plants to improve the productivity, to maintain the deterioration or deficiency of food and other needs caused by increase in population. That you can see in the strategies. On the next lesson you can see in detail plant breeding, the need of plant breeding in the science or in agriculture. See here, suppose two plants. This is a plant. This is another plant, and the leaves, same species, but the leaves are entirely different. Same species, two different kinds of plants, species same, producing beautiful flowers. Two plants of the same species, pollination takes place between these. That is heterogamy. What is heterogamy? The cross-pollination or allogamy or xenogamy, the transfer of pollen grains from anther to the stigma, from pollen grains from the anther to the stigma of another, stigma of the flower of another plant of the same species. Species same. One is genetically different, but species same. But the pollination between two plants of the same species, that is called allogamy or cross-pollination. See, listen. Cross-pollination, allogamy or xenogamy is the transfer of pollen grains from anther to the stigma of another plant. Stigma of the flower of another plant of the same species is called allogamy. Is called xenogamy or allogamy or cross-pollination. See once again, so simple. Pollination. Transfer of pollen grains from anther to the stigma is called pollination. Pollination is of two types, self-pollination and cross-pollination. See, self-pollination. The transfer of pollen grains from anther to the stigma of the same flower or that of another flower of the same plant is called self-pollination. Self-pollination is of two types, autogamy and heterogamy. Pollination in the same flower is autogamy. Pollination in different flowers of the same plant is allogamy, cross-pollination. Transfer of pollen grains from anther to the stigma of the same flower or transfer of pollen grains from anther to the stigma of the flower of another plant of the same species is called here an agent is strictly needed. Here agent is strictly needed, otherwise there is no chance for cross-pollination. So maybe insects, birds, ants, man artificial pollination, man or snakes, snails, then water, air, which are all the mediators of cross-pollination. See, remember please, the agent-oriented self-pollination is heterogamy, but agent strictly needed in cross-pollination because transferring the pollen grains from one flower to the another flower of another plant. So that is why. So the transfer of pollen grains from anther to the stigma of the flower of another plant of the same species, plant may be genetically different, is called xenogamy or allogamy or cross-pollination. See, here an agent is strictly needed, and so that is about general account of pollination. Then Animals, insects, birds, ants, snails, snakes, bats, winds, they are abiotic. Zoophily, entomophily, ornithophily, myrmetophily, malacophily, ophiophily, chiropterophily, anemophily, hydrophily. Such flavids are Suphophilus, Entomophilus, Ornithophilus. Myrmetophilus. See, agents and types of pollination and flowers. Animals, insects, birds and snails, snakes, bats, wind, water. These are abiotic, these are biotic agents. Rest on the next class. Okay.